morning to everybody. So today we will discuss on protein secondary structures and protein secondary structure prediction methods, and how we can actually have the secondary structures of proteins predicted using computational methods. More than that, how to actually understand the protein secondary structures and how we can work on different secondary structure prediction methods. So we'll have actually two slides, two different presentations. This is the first part of the presentation. So let's see. So this is the topic of today's presentation, that is protein secondary structure prediction part 1. Now, the first high resolution structure of a protein was first shown by Max Perez, John Cantu of Cambridge. So that was in 1958, and that was the 1962 Nobel Prize in Chemistry when the first high resolution structure of the protein myoglobin was presented. And say after 50 years, 60 years down the line, we have close to in 2012 there were around 80,000 structures, and till date we have around 110,000 protein structures in the structure database, which is called the PTB, protein structure database. And still, like, there are a huge number of proteins for which we have the sequence information, but we do not have the structural information. And if you go to the UniProt database, you have 500,000, 800,000 proteins available, for which not for which we have structural information available. So protein structures play a huge role. So there is a, as we discussed, like there is an increasing sequence to structure. What do you mean by that? We have this sequence information, which is most popularly represented as one-dimensional arrays of amino acids. There are 20 different types of amino acids that we have already discussed, and they can be represented as different types of structural units, like in terms of secondary structures, we have the helical structures, beta strands, and turns, and some coils. So prediction of three-dimensional structures from sequence is often a hard problem, and even sometimes very difficult. So we focus in this particular lecture like how we can have the secondary structure of the proteins predicted with reasonably higher precision, and we get significant advantage in terms of the functionalities of the proteins, because the structures are better conserved and they eventually relate to the functional association of those proteins. So what do you mean by a secondary structure? Secondary structures are the building blocks of the protein structure. So whenever we see a general protein and their 3D shapes, essentially they can be broken down to some kind of, most popularly seen as like some kind of helical structures, or maybe some kind of sheets. So these are called the ribbon diagrams that you see on the right. And on the left, you have the more like the backbone, like carbon-carbon backbone diagram. So if you see a ribbon diagram, then you will probably see either a helical kind of structure or a sheet-like of structure, and they are connected by small lines. So these helical structures are called often called alpha helix, and they are called the building blocks of the proteins. So as we said, there are 3 4 categorizations, but in to be on the most generic term, we have the helix. Most popular form of helix, which is the beta strand. So as you can see, they can also be numbered, as these are also directionals. Like on the right, they have number 1 strand, number 2 strand, and number 3 strand. So these are kind of strand sheet-like structures. So they are more like planar way of structures. So we call them as sheet, and the helix is like alpha helix is like they are mm, helical loops. And we have the third type of shapes, which are called turn, where you have a uh, sharp turn of amino acid sequences. And there is also a segment which is called a loop, which are basically interconnecting segments. Like in between sheets and the helix, you can have some interconnecting segments which are often called as loops and turns. In some terminologies, they are also called as coil, and in many cases they are also ignored. So you can see that most prominently you have those two structures like alpha helix and beta strands available as secondary structures. So if we see the helix, a bit more details about helix, so you can see that in 1951, Pauling he first invented the helix structure in a protein, and consecutive stretch of 5 to 40 amino acids. In general, say average 10 amino acids, they form a helical loop. Right-handed spiral conformation is usually the structure, and that signifies alpha helix, and usually 3.6 amino acids per turn. And you can see that there are dotted lines which are nothing but hydrogen bonds, and how these hydrogen bonds are actually formed, that is most important. Like even in sheets, you will see that there are hydrogen bonds. In a helix, there are hydrogen bonds, and these formation of hydrogen bonds allow the structure to take a specific form, in this case in a helical form. And the in between two corresponding heavy carbons in a helical shape, you can see that there is a distance of approximately 5.6 angstrom. So these characteristics make it alpha helix. There are certain variations, but in general, this alpha helix is the most common structure available in most of the proteins that we see today. And there is also beta strand. Also, it was invented in 1951, and it's also a polypeptide chain, also called as beta strand, range 5 to 10 amino acids, and the chains are connected together to form sheets. So you have different strands, and together they form a sheet-like of structure. And as you can see that there is a there are also hydrogen bonds among them. So this is in general form a planar kind of sheet-like of sheet-like structure, and strengthened by even here also we have strong hydrogen bonding that make this kind of sheets stable structure. So in general, the alpha helix and the beta strand, these two are the most popular structures available. And there are also like loops that connect secondary structures like alpha helix and beta strands and have various lengths and shapes. Sometimes they are also called as the coils, like any interconnecting string of amino acids. So this is also like sometimes there are a lot of research and a lot of uncertainties about what are the actual purpose of these segments. In many cases, they form the major part of the protein structure, and still they are often undefined in terms of their specific functionality. So when we do functional annotations, we mostly concentrate on the segments that have either the helical structures or the sheet-like structures. Okay, and it's really interesting. Like you can see that there are different secondary structures that are actually formed together, that are combined together or bound together to form a tertiary structures of a protein. And you can see that when we have a complete stable protein and there are different structural units, they get connected to give the protein a unique shape, and based on that particular shape the protein behaves in a particular way. And there are also like similar structures, like the same sequence having different structures, or similar type of structures, but they are not homologs. So many different varieties are there, and there are many different combinations which are available, and this makes the total study of the structural bioinformatics so interesting. And we will actually cover some parts of that in this particular course. As you can see that retinal binding protein and there is a globin. These are two examples we are showing here, and these are the tertiary structures, and you can see the secondary structure is shown here. You can see the primary sequence is given in the form of one-dimensional character string at the bottom, and then you have those these long arrows are nothing but beta sheets, and those ups and downs are called the strands. And then these small stuff, these are actually turned, and you can see that the annotations are also given. These are the extended strand, this is turned, this is like diffusible bonds, and this is like alpha helix, and this is like 3.10 helix. So different type of helix. This is called a pi helix. Okay, so in general, like whenever you have ups and downs, this is called helical structures. And the others, like the grayed out residues, have no structural information. So that is a significant part, as you can see, that do not have a significant structural information. In some cases, you can see there are more structural information and less grayed out parts. Just like in case of a globin, you have a lot of helix and absolutely no beta sheet. Okay, so there are different types of proteins can also be categorized based on the structures they have. Okay, so for example, this is only a protein which is having only helical structures, and there is absolutely no sheet. Okay, so there is a turn, and there are several turns, and there are only the helix. So in general, we have the secondary structure information, and we can actually have information. So this is a general information. And what do you mean by protein secondary structure prediction? And like when we have the sequence information, the sequence, as we mentioned just few slides back, that there are 500,000 to 800,000, even more sequence information is available in the UniProt database, and there are only around 100,000 or like very less amount of structural information, like tertiary structure information is available. So this gives a huge problem. Like it is computationally very expensive, as well as biologically extremely challenging to validate or extract the tertiary structure of a protein. So what is the way out? Like in many cases, we really do not have the tertiary structure information available. So the solution is, when we have the sequence information, we try to predict the secondary structure. What can be the possible secondary structure, so that we know like what are the like when we have a helical structure or something, then we can see that these are the functional units. So from there we can probably infer some functionality as well. We can predict some tertiary structural information. Many options are available. So secondary structure information from primary sequence is absolutely important when we do not have a readily available tertiary structure. So how do the secondary and tertiary structures relate to primary protein sequences? So there are so much ambiguity involved here, and sometimes it keeps researchers engaged and baffled that uh, how they are actually related. Like when we had the initial structures discovered, so we found that early experiments showed that sequence of information is enough to predict the structure, but eventually that is not true. Like it's true that protein structures are more conserved than protein sequence are more closely related to the function, but it's not always we can actually extract a secondary tertiary structure information from the sequence. Now, they have said like, say, different amino acid sequences determine similar protein structures. So, in a study in 1980, they have shown that completely two different sequences can actually have similar protein structure. So here we are showing some globins are like very most popular and very well studied protein family, like the hemoglobins in human, and there are is a myoglobin, and there are many different globins that actually carry oxygen in an organism. So there are significant number of proteins from the globin family which more or less do the same function, from the human to horse to like across different organisms. But you can see that the conservation is not much there, so that they are often significantly different. It's not always conserved. And in some cases, you can also see that the different sequences can also result in same structure, like even in case of completely different sequences which are not homologous, then they can also result in similar structure. So both these findings have been reported. So you can see that these two protein sequences are completely different, and however, they have very similar, as you can see that, in terms of their structural information. So this is actually very interesting observation, and these sometimes say that we can actually have it's very complicated to relate the sequence information directly with the tertiary structure of a protein. So how we relate the sequence and the structures. Like for example, in globin family, we can see that there are parts of the conserved so these regions are really conserved very well. So we need to sometimes see like what are these regions, and what is this like proline, why proline is conserved across this across the globin family, even if the structures are completely different. So all these queries sometimes come into picture, and like also it is important to see where are the gaps, like when there are these sequences, like where there are gaps, so these gaps are also important. So these are like, you know, the non-aligned regions and where these gaps are existing. So these are usually mapped in the loop region. That means they usually for example, in case of alpha helix or beta strand. So if we have pairwise alignment, so you can see that these gaps are primarily mapped into the loop regions. Even we can see that two remote homologs who are not related to connected, they can also map to the same structures, like RBP and beta lactoglobulin. Globulin, so that they can have very low identity. Like you can see that only 24% identity and 40% positives, RBP and beta lactoglobulin, but they have a very similar structure in terms of their like at least the visual alignment says that they have, like you can see that there is a strong helix and there is a beta sheet here and another set of beta sheets there. So they have a very similar structure, even if they have a very diverse sequence information. So keeping all this information in mind, so people started to focus more on like the study of protein secondary structure prediction gained more momentum, and people started to see that given a sequence information and giving the variation of, you know, like structure to secondary structure to tertiary structure, like primary sequence to tertiary structure variability, it is probably more meaningful to have a secondary structure information extracted for protein function prediction or to assess the functionality of a specific protein. So what is the basic question? So the basic question is here that given a primary sequence like this, ADSGHY, what is the secondary structure it will adapt? Whether ADS is going to be represented as a corresponding alpha helix or beta strand or a turn or a loop, which is often called a random coil, which is in interconnecting loops. So this is the basic question, and this is often considered as one of the classical problems in bioinformatics, where we have a sequence information which is abundant in nature, and as we discussed several times, that you can see that the sequence information can actually be extracted directly from the genetic sequence, and after the transcription, and you can see that there is a direct 1:1 mapping, considering even if there are certain variations to splicing and other stuff, but still you can have a more or less direct mapping from a genetic code to a primary sequence. But from sequence to structure, there is a huge variability. So we really do not have much idea how it actually map to a specific. There is no rule that a sequence of amino acids will always form a helical shape or a beta strand. So this gives room to computational analysis. Okay, so that is the objective of this course. And we usually apply different types of statistical methods based on amino acid frequency. So traditionally, people started to look into different types of, the frequencies of amino acids, how they can be transformed to, what is the probability of a particular amino acid to get transformed to a helical shape or a beta strand. So this was popularly studied, and then there were different machine learning methods like support vector machine, neural networks, everything. These were applied. Hidden Markov model was very popular, and we will also study some hidden Markov model approaches in this particular course. One of the first approaches proposed was by Chow and Fussman, which was in 1974. And what they wanted to do, they wanted to say that the propensity of an amino acid to be part of a certain secondary structure, for example, proline, as you can see here, has a very low propensity to become an alpha helix. Okay, so you can see that the proline is having a very low propensity. So what they did, they considered certain amount of protein structures that were available to that date. In 74, only a handful of protein structures were biologically validated or experimentally validated. So they took out all those structures and they just did a count or statistical analysis that how many alanines were actually converted to a helix. A stands for P of A is propensity of alpha helix, propensity of beta sheet, or propensity of being a turn. So they considered all of them, and as you can see that in certain cases you can say something, right? Say for example, in case of say tyrosine, it is having more propensity to form a beta sheet than alpha helix, or maybe a turn as well. Similarly, if you see that glutamic acid is having a significantly high propensity to become an alpha helix than a beta sheet. So he was working on this. For example, asparagine is having a very high propensity to become a turn, just like glycine. So these were his observations, and he started to build a model on that. And initially, the rate was very not really good, but it was actually the first method on which they started on. And there are a lot of advancements even on the basis of Chow-Fussman's method, and there were a lot of approaches that came on the advanced stuff. And even today, the propensity of an amino acid is still considered as one of the features, even while using advanced machine learning approaches. Like how we can do it, like it's sometimes very it's not easy to predict about a specific amino acid and its corresponding secondary structure. As you can see that when we talk about alpha helix, there are usually 5 to 10 residues in continuous. Like most alpha helices are around 12 residues long. Most beta strands are around 6 residues long. So if we just try to predict alpha helix and beta sheets, so if you find just one amino acid in between which is not a helix, well, the other ones are predicted as helix, then probably it is a outlier, right? So we try to work on a neighborhood based approach. So people started working on a neighborhood based approach, and we call this as a sliding window. So people started working on sliding window sizes of 6 to 12, and there are many things, and calculate a score of each window. If it goes beyond the threshold, then predict whether it is alpha helix or a beta sheet. So you are predicting not as just as a single amino acid, to be helix or strand. Rather, you are actually predicting for the whole lot, or sometimes not even the whole lot. You are often predicting just for the central amino acid, just for the central amino acid in the overall window. But in this case, the original Chow-Fussman's method, they said that okay, so if you have a uh, sliding window which moves like this, and you slowly move on, and then you just estimate that what is the threshold, what is the combined score for being alpha helix or beta sheet, and based on that, if it crosses a particular threshold, then you call the whole stuff as a um, whole window as a helix or sheet. Otherwise, you stop there. So this was the approach adopted by Chow. Now, see, these are like these were very old and initial approaches, and at those days means at those times there were not really advanced machine learning approaches available, and not really a lot of sequence information, structural information available. Also important was like multiple sequence alignment. How could we associate the conservation information? As you can see that there are local alignments of multiple sequences. So if you can actually work with a group of closely linked sequences, then there is a very high chance that you can actually predict structural information of a sequence. Like you have a sequence information which matches in terms of sequence 90% with 10 other sequences for which you know the structure. So we can actually look at the conserved regions and look at the structure in those conserved regions. So this can be done in multiple sequence alignment, and we can extract different types of scores. For example, uh, PSSM scores, and many other stuff we'll very quickly discuss. And there are other algorithms, different machine learning algorithms and hidden Markov models that also we will discuss. And success rates slowly move to around up to around 80%, and even today we have 85-90% close to that accuracy we can see. And that we'll discuss in the next class, like what are the state-of-the-art approaches. So many machine learning approaches today use secondary structures like using multiple sequence alignment. So methods like PhD, PSI PRED, we'll briefly discuss about them in a subsequent lecture. They use different types of machine learning approaches. So what is the basic approach of those type of algorithms? So it says that we have a big database, and the first step, like you have a query sequence. So you have a sequence information available for which you do not know the structure, and you want to predict the secondary structure of the query protein. The first step is the generate a multiple sequence alignment. So you probe the protein in the database, and you find all the set of proteins that have a significant alignment with the query protein. So that is called multiple sequence alignment. So in this example, suppose you have a query protein and you obtain 4 proteins that are significantly aligned to the query protein. So you get a multiple sequence alignment score. So what you can do, like you can actually have a iterative approach as well. Like you can have a second set of query based on the proteins that you get from the first iteration, and finally you can get a whole lot of proteins representing a family of proteins using multiple sequence alignment. And then these, you know, like the sequence profile of this protein family can be compared, and you can use this approach or use these known sequences informations to predict the unknown structure using a machine learning approach. A very simple method that people use is position specific scoring metrics. So that we will discuss in a subsequent class, where you can very easily say that, okay, you have 100 proteins available with respect to the query sequence that match, say, at least 80% of the alignment score. Based on that alignment score, you actually extracted a protein family, and then you can essentially extract a, if you have a uh, query sequence of length n, you can actually extract a 20 cross n matrix, PSSM matrix, from the query sequence using the multiple sequence alignment. And you can just train using any standard, say for example machine learning approach, for example support vector machine is very popular, and you can use some kind of windowing approach. Maybe you can use a 9 window, or say maybe a 7 window structure to predict the central residue of the uh, sliding window using the PSSM matrix. So in general, this is just an overview that how we can use a sequence profile of the protein family and extract some kind of meaningful information from them. Okay, so this can be done using multiple sequence alignment, and there are many methods that are actually following this, and these are very standard. And we will probably discuss some of those methods in greater details in subsequent class. But here, our objective is just to understand the basic logic. Okay. Now, let us move on to hidden Markov model, which is also a very popular method, and for prediction of protein secondary structure, this has also been used classically. Okay, so although we are a bit out of scope here to study hidden Markov model, but I'll very quickly browse through the architecture of hidden Markov model, and how it can be used to solve some kind of protein secondary structure prediction task. Okay, so what is HMM? HMM is basically a statistical model and well suited for many tasks in molecular biology, and not just in molecular biology. It can be used in even like natural language processing and many other text processing stuff. Okay, so it generates a probabilistic profile from a family of proteins searching for a database or other members of the family. So resemble the profile and weight matrix. So it generates a weight matrix. So we'll see how it means, and then it can also be used for grammatical structures like gene finding, recognize signals, prediction, like different types of like what can be the gene, the function predictions, etc. So we can follow the specific rules of a gene, and we can apply it in some kind of probabilistic and predictive models. So we'll specifically see how it can be applied there. But before that, let us assume a very simple, coin toss game. And if we see that, suppose we have a fair coin and a biased coin, and question is like, is it possible to determine whether a biased coin has been used based on the output sequence of the head and tail? Suppose we have a sequence of head and tail, and can we say that the coin is actually biased, or whether there is actually a fair coin or a biased coin is used? Like for example, sometimes we see that some of the in the game of cricket, sometimes some captains lose a sequence of tosses. Okay, so sometimes we find we think like, oh, is it like is it like, is it like really a fair situation? Are there any kind of bias? So you can probably use a hidden Markov model there and check it out whether there was a really a fair coin used in this kind of situation. So what is the situation in a fair coin toss? You can have it is everywhere it is zero point 0.5. So you can have you can design a simple Markov model. uh, there can be just two states, like one can be head and there can be tail, and there is there can be two possibilities, right? When there is a head, what is the probability that the next state will be tail? So it is zero point 0.5. And what is the probability that the next state will be head? This is again 0 0.5. And if it is a tail, again it is an equal probability. So you can either get a tail in the next toss, or it can also get a tail in the head in the next toss. So again, this is a equal probability. So it is a fair coin toss, and you have equal probability both for head as well as a tail. Now, what if there is a biased coin? Now, for example, in case of a fair coin, you have two possibilities, head and tail, equally having 0.5, 0.5, and if it is a there is also a 50% probability that it stays in the fair coin state and goes to the biased coin state. And in case of a biased coin, you have say you have 90% probability of staying in the biased state, 10% probability to go to the fair state, and there is a 90% probability to generate this generate it as a tail, and 0.1% probability of head. Now, visible state do not correspond to the hidden state. So these are only the visible state. So you can see that this is the visible state. So can we say that which coin has been used? So this is the generic problem that we try to handle in case of a hidden Markov model. So given these sequences, can we say whether this is a biased coin or a fair coin? For example, in case of a in case of a weather prediction problem, suppose we have some probability of being sunny, some probability of being cloudy, and some probability of being rainy. Okay, so what is the sequence of events of wearing a sunglass, taking an umbrella, or taking a cold weather gloves? Okay, so this is basically snowfall. So what you observe, these are the observation probabilities, and these are the hidden states. So based on these, so can we model this? So what are the ingredients of an HMM? So the ingredients of an HMM are like what we have. We have some collection of states. For example, you have the n number of states, S1, S2, S3 up to Sn. You have n number of states. And you have state transition probabilities. State transition probabilities means that given in state Qt with Sj, what is the probability that you will land into state Qt plus 1? Okay. And given in a specific state, for example, being in a sunny state, what is the probability of going to a cloudy state or rainy state? Okay, so that will be different from being in a snowy state to a rainy state. So what is your current state Sj, and what is your current state Si? Okay, so we will take up a specific problem, and then we will see that how this state transition probability, this is basically a matrix that can be constructed. Similarly, we have the initial state distribution, so that what is the first situation of the initial state. Like suppose you are predicting for the whole month of say September, and the first day of September, what is the probability of the of it being rainy or something? Okay, so this gives the initial state distribution. Once you know the probability distribution of the first state, then you can map it to the state transition. Like what is the next state? Okay, so first state distribution is what is the probability of a state to remain in say Si. So that is pi I is the initial state distribution. And what are the observation probabilities? So what are the different types of observations that you can have? Like suppose you can have like, okay, someone is taking umbrella, someone is taking gloves. So these are all observations. Like there can be m number of observations, which is independent from the n number of states. Okay, and what are the different observation probabilities? That given Qt equals to Sj, what is the observation of OK? So we will actually work with these 5 parameters, that the collection of state transition probabilities, initial state distribution, observations and observation probabilities. With this, we can actually formulate any kind of problem using hidden Markov model. We will immediately go to, like it shows something like this. Like if we have states like, like sunny, rainy and snowy, and these are the state transition probabilities, that what can be the transition of state A to state B and state B to state C. So this can be formulated as state transition probabilities. What are the initial state probabilities? What is the in the first case, what is the probability of being sunny, which is 0.7? What is the probability of being rainy is 0.25. What is the probability of being snowy is 0.05. So this is the initial state probability. And what are the observations? There can be m different types of observations, and there can be m different types of m cross m type of observation probabilities. Okay, so we will directly go into, instead of taking the example of like weather prediction problem, we will directly go into the problem where we can actually see that how a secondary structure can be predicted. So in general, typical prediction problems are like annotations, like classifications, consensus, and training. So as I said, like hidden Markov model is more a pattern classification or pattern recognition topic. So we will see how we can actually use hidden Markov model to solve a problem in case of protein secondary structure. For example, like if we have a sequence of like TG, TA, GPO, primary sequence, what is the probability that the secondary structure will be like this? Okay, so HHH loop, and this is beta strands or sheets. So helix, loop, and beta strands. So if this is the sequence, what will be the probability of this? Okay, so we can first formulate the model, hidden Markov model, and then we can try to see what is the estimated probability. Now you see how many sheets are there, how many states are there. So we have 3 different states. So one state is like any amino acid can be a alpha helix, it can be a beta sheet, or it can be a turn. So there are only 3 possible states. So we have a state transition matrix. So we will see how we got that. So first thing was in number of states. So we have number of states equals to 3. So we have a state transition probability. Then what we need? We need the observations. What are the different observations? Like, see, we have 20 amino acids, right? So every amino acid can be like we studied in Chow-Fussman, and Chow-Fussman said that every amino acid can take a specific propensity. So for example, alanine can have a 6% probability to become alpha helix. It has 4% probability to become beta sheet. It has a 3% probability to become turn. So these are the observation. There are 20 different observations, and there are 20 different observations. Okay. So what do you mean by state transition? State transition means, suppose an amino acid has been classified as a helix, and then what is the probability that the next amino acid is also helix? We see that this is the state transition probability of 0.8. So we have the 80% probability of a helix to become helix in the next stage or next state. Okay, our transition state transition probability of helix is 0.8. Now helix to beta sheet is 0.05. That is, so 5% probability of helix to be immediately the next one to be beta sheet is only 5%. But helix to turn is 15%. So once it is helix, the next state becoming turn is 15%. So this is state transition probability. Okay, so let's see again what it says. The probability of observing alanine as part of beta sheet is 0.05. Okay. The probability of observing a residue which belongs to alpha helix followed by a residue belonging to a turn is 0.15. That is 15%. And alpha helix followed by alpha helix is 80%. Now, suppose we have a sequence. What is the probability that the sequence starts with an alpha helix? That is the initial state probability. So for the first time, this is for the first time it is entering a state. Okay, so that is the first character or the first symbol in the sequence. So what is the probability of that to be alpha helix? So we can estimate that. Again, from what do we estimate this? Again, we estimated this just like Chow-Fussman said. Chow-Fussman assessed a lot of known structures. Using multiple sequence alignment, we aligned many known sequences. So just like that, from the known database, we can actually start with a specific we can start with a sequence with a specific structure, like for example helix as 0.45. Similarly, the initial state probability of being a beta sheet is 0.45. and say for example for being a turn is really low because it's really unlikely that it starts with a turn, so let us give it as 0.1. Okay, so for example, what is the probability of sequence TGQ will be a helical structure? This is a question. Suppose if I give this question as an exercise, and if I give this particular transition matrix, so can you say that, can you estimate that given TGQ to be HHH, what is the probability? Let us estimate. So what do we have for T? We have first T to be H, we need to have a initial state probability. So what is T is the first character, so initial state probability of T to become H is where this is the initial state probability of any character to be helix. Okay, so we have 0.45. Then what is the observation probability of T to be helix? Let's see again. In T, see it is 0.041. The observation probability of T to be helix is 0.041. So we multiply the observation probability with the initial state probability for T, and it is 0.45 into 0.041. So this is about T. Now what we are suggesting? H and H. That means you are estimating for helix, and then in the next step also you are trying to remain in helix. So what it means that the previous state T was in alpha helix, and again you are estimating the probability of it becoming a alpha helix again. So what is the probability or state transition probability of helix to be a helix is 0.8. So we are next is helix to be a helix in the next state is 80% probability, and this is G. G to be a helix, where is G? G to be a helix is 0.28, 0.028. So we multiply 0.8 with 0.028. That is the observation probability of G to be helix. Now again, helix to helix is again 0.8. Okay, so for Q we have estimated this, and from where this comes, like the observation probability of Q must be, observation probability of Q is 0.0635. So 0.0635. So for these three amino acid sequences to be HHH, the probability is 0.0020995. So we can just product. Suppose if I give an question that what is the probability of TGQ to become H, H and S. So if I give TGQ and HHS, then how can you estimate? So H, H, and then for S, these two will change. So in such case, you'll have to estimate what is the transition probability of helix to beta sheet, and in that case, this will be 0.05, and Q to be S is 0.04. So instead of this, so it will be 0.05 and 0.04. So you can estimate another value. So this is how you can actually estimate the observation probability. For example, the way we shown here. Say, for example, you have a sequence this, and what is the sequence probability of a sequence to be like this? We can estimate that. And there are many different ways how we do that, and there are different optimization algorithms like Viterbi optimization, Viterbi algorithm, so that you find the optimum. You actually need to estimate all the possibilities. and then you estimate the optimum path, so that you get the cost becomes the probability score is actually maximum. So in each step you actually have HLB, 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 and you actually find the optimal path between that. So that can be done using fitted B algorithm. So that way, you with the known state transition, observation probability, and initial probability matrices, you can use hidden Markov model to predict secondary structure from a given sequence. So this is what we have learned today, and that is the part we have covered. And so here in this lecture we have studied how protein secondary structure can be predicted, what you mean by protein secondary structures, and especially how very classical methods, for example, Chow-Fussman method for propensity or statistical analysis, and some kind of window-based assessment, and then how we can actually use hidden Markov model to predict protein secondary structures, that we have studied. And in the next lecture we'll see how we can actually how different state-of-the-art methods, like we mentioned, like PSI PRED, PhD, what they use. For example, they extensively use multiple sequence alignment and position specific scoring matrix, and then with that they use different types of machine learning methods like support vector machine and all. So all these things we'll actually check in the next class. Thank you very much.